Assalamualaikum and hello world. Cerita dah macam ni.
BI.
Oh, sorry. The story starts from the bottom field. In 2010, Gerald Ball, an American inventor, designed a project to produce biofuel from the little creature called algae. He put them inside a somewhat container so the biofuel can be harvested. So, what exactly is the container? The answer is, the container is what we call bioreactor. And yet, where on earth is the intro for this video?
Welcome to Gloria.
Okay, bioreactor is referred to any manufactured or engineered device or system that support a biologically active environment. In other words, bioreactor is a vessel in which a chemical process carried out which involves organism or biochemically active substance derived from such organism. This process can be aerobic or anaerobic. There are many bioreactors out there. Here are some of them. We have batch bioreactor, which is majority used, maybe because due to the low cost and the flexibility with varying product offered. The second one is continuous, which is the culture medium of microorganism is continuously fed into the bioreactor, thus result in continuous productivity and output. We also have fed batch bioreactor. As the name goes, fed, that means the bioreactor is fed. Nutrients are added to the bioreactor if the bioreactor approach depletion of nutrients. We also have trickle bed bioreactor, which widely used for aerobic wastewater treatment and extensively used in processing plant. Back to our American Gerald Ball. What exactly the type of bioreactor that he used? Actually, he used photobioreactor to grow the algae. This bioreactor incorporated with some type of light sources, so it can be used on another small phototropic organism such as cyanobacteria and mosaic.
Okay, next for the basic compartment and the basic design of the bioreactor. Bioreactor mainly consists of 5 major compartments, which is agitator, buffer, sparger, jacket and also controller. Okay, for the first one is the agitator. Agitator is a compartment that looks like a fan which spin the culture media, which is contain nutrient, cultivation cell and also dissolve oxygen and other things to make it a homogeneous media. For the second one is a buffer. Buffer is a compartment where it break the vortex formation that make by the agitator earlier to avoid the overuse of the energy. For the third one is a sparger. Sparger is a compartment which supply oxygen to the culture media and also help the agitator to make the media mix well. It also avoid sedimentation at the bottom of the bioreactor. The fourth compartment, that is jacket, which is outer layer of the bioreactor that function to keep the bioreactor temperature at a constant level. For the last major compartment, which is controller and sensor that using a probe, the first one is the speed controller, which control the agitator spinning. The second one is the temperature, which maintain the temperature by lowering and raising the temperature, reaching the optimum temperature desired by the cell culture. The third one is the pH controller, which maintain the basicity or acidity of the bioreactor. The fourth one is the dissolved oxygen, which control the bubble formation if the dissolved oxygen at high level or control the fermentation that produce if the oxygen level is lower. The last one is the foam controller, which avoid the fermentation particle produced by putting anti-foam substances inside the bioreactor.
Well, hello there world. Now let's move to importance of bioreactor. I'm very sure that you all already know who is the Louis Pasteur, right? Based on Pasteur work on fermentation, Koch laid the foundation of the bioreactor as we use today. As a result, the scope of bioengineering has grown from a simple bottle microbiology to the industrialization of the not only beer, wine, cheese, and meat production, but also the production of biotechnology new products such as antibiotic, enzyme, hormone, vitamin, sugar, and other also can be built. Here we can see the importance of the bioreactor. Why bioreactor is better than traditional chemical reactor? Actually, bioreactor is different from traditional chemical reactor because they support and control biological entities, which is bioreactor system was designed to provide higher degree of control over the process of upset and also contamination. Since the organisms are less stable than chemical, so if we not control well, it will mutate, which may alter the bioreaction and also the physical properties of the organism. So here we can see that bioreactor will maintain the desired biological activity and eliminating undesired activity. Other key difference between chemical reactor is selectivity. Higher selectivity in bioreactor, there is measure of the system capability for producing preferred product instead of other outcome. In fact, selectivity is especially important in production relative complex molecules such as antibiotics, steroid, vitamin, and also protein. For example, if we want to make products such as antibiotic, which is penicillin, we just can use penicillium chrysogenome species to produce this penicillin antibiotic. Other important is, although the molecular structure of penicillin is known, it may be synthesized by chemical method, but it's not economic to do so. The production still relies on fungal fermentation based on biological principle. This is because the modern strain are more productive compared to the early strain.
Assalamualaikum warahmatullahi wabarakatuh and hello. How is it going everybody? I hope you guys in a good mood. Let's jump straight to the action. Okay, today I'm going to talk about the example of bioreactor in food industry. Okay, for your information, bioreactor involved in many industries such as photobioreactor, medicine production, sewage treatment, NASA tissue cloning bioreactor, and food industry. However, in this video, I will just go through about the example of bioreactor in food industry. Okay, as Hafiz explained earlier, there are many types of bioreactor. Okay, the bioreactor that I will explain in the food industry is three types of bioreactor, which is first is batch bioreactor, second is fed batch bioreactor, and third is the continuously bioreactor. For these three types of bioreactor, will produce different kinds of food. For the first bioreactor, which is the batch bioreactor, in the food processing industry, yogurt, beer, and soy sauce are made up of batch bioreactor. I will take one example, which is beer. A batch bioreactor is filled with food medium and then inoculate. Then the fermentation occurs. So this is how the beer is produced. The next bioreactor is feed batch bioreactor. The food that use this bioreactor is vinegar. When making the vinegar, low levels of ethanol must be maintained because high levels are toxic to the bacterial cells. For this reason, the ethanol is trickled into the bioreactor slowly. So this is how the vinegar is produced by using the feed batch bioreactor. Okay, then we go to the last bioreactor, which is continuously bioreactor. The fresh medium is continuously added into the bioreactor, and at the same time, the bioreactor fluid is continuously removed. The cells continuously propagate on the fresh medium entering the reactor, and at the same time, the metabolic waste products and the cells are removed in the effluent. This causes the bacterial cells immobilize in the reactor to maximize their rentations and increase the productivity. Okay, the example of using this continuous bioreactor is the corn. Corn is the mycoprotein which well known in US.
Actually, bioreactors have major roles in many fields such as bioprocess, plant propagation, fermentation, biolandfill, or also in tissue engineering. Okay, I will explain a little bit advantages of bioreactors in landfill. Okay, first, bioreactor actually will help in order to reduce potential for our pollution. How this happen? This presumably comes from recirculating water that we call leachate within the landfill to collect toxic metals and other harmful substances rather than have such substances gradually get into the ground beneath the landfill. The second advantages of bioreactor technology actually significantly reduce the times required, for example, decomposition of waste. Or we can see the advantages in medicine, for example, in production of antibiotics. How this happen? This is done by using the continuous bioreactor, which is no need to be shut down, give a shorter turnaround time. The third advantages of bioreactor that I found in tissue engineering from one journal, bioreactor actually acts as a device that provides a controllable, mechanically active environment that can be used to study potentially improve engineering tissue structure, properties, and integration. This can be applied, for example, in tissue formation. Now we are enter the second part of my discussion, which is about disadvantages of bioreactor. First of all, bioreactor are demanding in terms of cost. Why I said so? Because initial cost of bioreactor are very higher. Because these devices are quite expensive compared to the traditional waste. The second disadvantages of bioreactor is it is designed is relatively complex engineering. Thus, whenever the optimum condition is not fulfilled, the cells or microorganisms that involved in the bioreactor are unable to perform the desired function. Hence, absolutely, it will affect our results. The third disadvantages that I can share to all of you that one example of disadvantages in bioreactor in landfill is bioreactor tend to produce more hydrogen sulfide H2S, which is smell like rotten eggs. I'm very sure that all of you know that hydrogen sulfide is quite very poisonous, corrosive, flammable, and explosive. If you have any question or comments, you can subscribe to our Facebook accounts or our Twitter accounts. And if you have any question, you can ask to us later. Thank you for watching.
There are so many types of many.
I already know that you and Memula
Helps in order to...
Okay, tak apa.
Testing.
Stop, whoa, yes, wait a minute, Mr. Postman.